Okay, to test the biochemical properties of our bacteria from our phone, I'm going to transfer some from the Petri plate into glucose fermentation. And you notice there's a little inverted tube in there called the Durham tube. That's to catch any gas that's produced. We'll also be able to see if acid is produced, it'll change from red to yellow. So that's glucose. Then I'm going to repeat it with sucrose, same thing. Lactose. So those three all look the same. Those are carbohydrate fermentation tests. Then I'm going to do a litmus milk. It starts purple, and we're interested in whether curds and whey can be made by the bacteria as they basically digest the milk proteins. And then finally, I've got nutrient gelatin, which is like Jell-O. So gelatin is a protein. I'm going to stick the bacteria in here, and we're going to see if they can digest gelatin. So for each one of these, sterilize the transfer loop, let it cool, take some bacteria from our Petri plate, just a little bit, open up the tube, stick the bacteria in, and just shake it around, and then immediately sterilize the transfer loop. And I'm going to repeat this for all of those tubes. The gelatin is pretty firm, so I'm going to stab it down into that solid nutrient gelatin, all the way down to the bottom. And always sterilize before and after. All right, now we're going to pour a Petri plate, but instead of nutrient agar, this is melted starch agar. So the active ingredient is starch. We're going to pour it, let it solidify, and when it solidifies, streak the bacteria and see if the bacteria can digest starch during incubation. Okay, our last biochemical test is going to be inoculating this starch agar Petri plate with a single line of the bacteria from my cell phone. So I sterilize my transfer loop, let it cool, take a little bit of the bacteria, open the starch Petri plate, and we're just going to do one streak. Okay, sterilize it, label it starch, and put it in the incubator. Okay, so here are the 5 biochemical test tubes, and then our starch agar Petri plate. They've all been inoculated with bacteria. Now we're going to incubate them overnight at human body temperature. Notice again, I've got the starch plate upside down so that the condensation on the lid doesn't drip onto the bacteria. All right, we'll see those results tomorrow. All right, let's see how our biochemical tests are. Here's our starch Petri plate, nice line of bacteria. Here are the test tubes. Let's take this back to the table and analyze it. Okay, the first thing I'm going to do is the Catalase test. Catalase is an enzyme that will create oxygen bubbles in the presence of hydrogen peroxide. So I need to sterilize my transfer loop, let it cool. I'm going to take some of the bacteria from my starch Petri plate and put it on the slide. Okay, don't need too much. Sterilize. And now we want to put just a little drop of hydrogen peroxide onto the slide and look for bubbles. Definitely some bubbles forming, so that's positive for catalase, and those are oxygen gas bubbles. The next test we're going to do is to see if the starch has been digested or hydrolyzed. So we're going to open the Petri dish. We've got iodine, which iodine plus starch will make a dark brownish black reaction. Going to saturate the Petri plate. So what we're looking for, if the starch has been digested, this will leave a yellow halo around the bacteria. Now, why don't we give that just a few minutes to react? We'll come back to that. Okay, our starch Petri plate has had time for the iodine to interact with whatever's left. Now look at our results. If starch were present, the starch and iodine would make dark brownish black. If the starch has been hydrolyzed or digested, it'll be yellow because there's no starch. I'd say this is yellow, and so it's hydrolysis, positive for starch hydrolysis. And let's look at our other tests. Now remember, these were all red. We've got glucose, lactose, sucrose. Lactose and sucrose are red, so those did not change. So let's look at glucose. Glucose turned yellow, and there's a little air bubble trapped in that Durham tube. So the yellow means acid was produced, and the little gas bubble means gas was produced as the bacteria metabolized glucose. As far as these two, those are going to be negative for acid and negative for gas. Okay, moving on. The litmus milk stayed purple. Now, if it had made curds and whey, it would have looked like this, or if it produced acid, it would have turned pink. So this is negative for the litmus milk reaction. And finally, nutrient gelatin. Remember, this was solid. Since incubating, it has turned liquid, which means the solid gelatin has been digested or hydrolyzed. So that's positive for hydrolysis. So to fill out our results in table 3, glucose was positive for acid, positive for gas. For sucrose, negative for acid, negative for gas. Lactose, negative for acid, negative for gas. Litmus milk was negative, so this is no acid, no curds, and no whey, Jose. Okay. Protein gelatin hydrolysis was positive for hydrolysis. Starch hydrolysis was positive for digestion hydrolysis. And the Catalase test was positive, so that created bubbles. So we're going to look at how to use these data, plus our other 2 tables of data, to identify our unknown bacteria. I'll do that in another video.